So far, so we have reviewed the concept of protein-ligand docking calculation. Then why is protein-ligand docking so difficult? So the first is that, as I mentioned, there's a sampling problem. So the sampling means that, so the protein and ligand, they both can have many possible structures. So the structure of proteins and ligands both are possible, are flexible, and the structures of protein may change, if ligands bind. Also, the ligands can have, multiple conformations. As I mentioned, the single bonds, because the single bonds are rotatable, right? So the second reason is that there's another type of problem called scoring problem. So the scoring means that so whether we can calculate free energies accurately, binding affinities accurately. But the truth is that it is hard to calculate an accurate binding free energy between proteins and ligands, because proteins and ligands consist of many atoms, right? So the proteins are generally consist of, thousands of atoms, and ligands may have, about 100 atoms sometimes. So they consist of many atoms. Actually, so in order to get, accurate binding energy between, two molecules, then we have to solve Schrödinger equations of a protein and protein ligand complex, But doing quantum calculation of such a large molecule takes forever. So it doesn't end. So we have to. So we cannot use. We cannot solve Schrödinger equation of a protein. But instead, so we need to use. Approximate function. We need to use. We need to do some approximation. We need to use approximate. Energy function. And another reason is that entropy is, so as I mentioned, so the accurate binding free energy is about the Gibbs free energy, right? But the Gibbs free energy is defined as a delta H minus T delta S, as you know. So here the delta S is entropy, right? Entropy of the state. Entropy of a state. But the entropy of a state is hard to calculate, because it is related to the number of possible states. So the entropy is conceptually, as you know, so the entropy is connected with the number of possible states, right? Number of possible conformations, and so enumerate, counting, how many states are possible for such a flexible molecule is very complicated problem. So entropy should also be measured, should also be estimated, approximately. Here, the approximation is also needed, okay? So because of these two, reasons, so the sampling and scoring problems, so the protein ligand docking is not a trivial problem, trivial task. So we need to, as I mentioned, in order to solve this problem approximately, so we need to introduce some assumptions. So we'll talk about some assumptions, and we'll talk, how real, researchers and programs are solving this problem. So the basic assumption of general protein ligand docking calculation is that the ligands may bind to structurally complementary sites. So the structurally complementary site means that, structurally they are, complement to each other. So which means that, so let's say that, so we have a protein looks like this on the right side. Let's say that we have a protein looks like this, and its binding, and let's say that its binding site is here. Then let's assume that, if our ligand looks like this, then it actually doesn't fit well to the shape of the binding site, right? So we can assume that it may not bind with this protein strongly, or this ligand will not bind at all. But if some ligand looks like this, then if it comes to here, then it fits almost perfectly, right? So it fits well. So like a Lego blocks, right? So when a ligand fits well to a protein, then we can assume that so this complex will interact very strongly. So this is kind of the structural complementary, okay? So they structurally match to each other so that they can bind strongly. Then what is the rationale of this assumption? So why? So the rationale, what is the rationale? So the rationale of this assumption is that for many protein ligand complexes, actually the van der Waals interactions are generally the most important, or sometimes one of the most important factors. But as you know, the van der Waals interactions, between every pair of atoms, van der Waals interactions exist, right? So therefore, van der Waals interactions becomes larger or become stronger as more and more atoms are involved, as more and more atoms are interacting each other, okay? So that's the basic rationale of structural complementary. So here the green surface. So here the green surface is the surface of protein. So the green is a protein surface, and as you can see, there's this ligand. This green ligand fits well to its binding site. Then how do we define binding site and structural complementary? So in order to identify structural complementary, so as I mentioned, as you've seen, so that we need to know, the surface. We need to know the shape of the surface of protein, okay? So, we've seen the protein structure with ribbon and atom representation, in the previous class, right? So this is the structure of a protein. But, if you think about, but here, so we're representing each atom as a stick, right? But if you think about the van der Waals radius, actually, each atom have van der Waals radius, right? Like this, right? So if you think about those, van der Waals radius, then the actual surface of this protein would look like this. So here this cloud-shaped thing. Okay, do you get the difference? So here, to make it clear, the atoms are represented as sticks, right? But here, if you think about the van der Waals radius, so this one become the actual surface of HIV protease, okay? So the van der Waals radius is considered. Okay, so in order to find the right, in order to identify right, binding site, and so we have to take a look at the surface of a protein. Okay. Then how do we generate? How do we, define protein surface? So the protein surface is sometimes called as accessible surface area. So which means that solvent atom can. So the area where solvent atom can access, where the solvent atom or solvent molecule can approach. And the algorithm to calculate this is called the Lee and Richardson algorithm. So this is also sometimes called as Lee and Richardson algorithm surface. And Lee is named after a Korean, very famous Korean protein scientist. Dr. Lee. So he is a retired. So he used to work. He had been working in NIH for many years, but now he is retired. But so his paper is very, well cited, and so he is a very famous protein scientist. So the basic idea, the idea is simple. So the basic idea is that roll a probe on a protein surface. So on the van der Waals surface. So let's say that these red dots. So let's say that these red dots are atoms from protein. This is an atom, and this is an atom from protein. Then we can think about its van der Waals radius of each atom, right? Van der Waals radius of each atom. Then let's think about this. Purple ball. So this purple ball is a water molecule. And if we roll this water molecule on the surface, on the atom of proteins, then we can get these dotted lines. So we can get these dotted lines by connecting where the center of this probe. So let's assume that the water molecule is spherical shaped, and let's call the center of, let's think about the center of this probe, and we can connect we can connect the paths where the center of probe follows, and this dotted line actually this dotted surface becomes the accessible surface, because so this water molecule, because this probe cannot approach closer than the van der Waals radius, right? So this dotted line forms the accessible surface. Okay, so this is the basic idea of forming and defining the surface of a protein. Okay. So when surfaces are generated, so actually so you can generate protein surface. You can visualize protein surface using visualization softwares like Chimera or VMD. So if you draw protein surfaces, so this is the protein surface, then you can see that some parts are some parts are concave and some parts are convex. So the convex, so if you're not sure, so the convex is an acronym. Convex is the opposite of concave. Okay, so you can see that some parts are concave, right? Or you can see that some parts are like caves, right? But if you think, then those concave parts must be better for ligand binding, right? So if you think about some surface is concave, then the ligand comes in, then they both can have many interactions, right? Because so it is surrounded with surface. But if some ligand approaches to a convex surface, then so it may not have many interactions, because so with the convex surface, the molecule is hard to be attached, right? So hard to be accommodated, embedded, right? So usually, as you can see from here and here and here, so usually these concave sites of a protein surface tend to be a binding site of a ligand. So in other words, ligands generally bind to concave sites of a protein surface. Okay. So the ligands are generally, bound to those concave sites. Then how do we find? So if we don't know the binding pocket or the binding site, so here the pocket means binding site, then how do you find, binding pockets? So if we know the geometry of the protein surface, then binding pocket, possible binding pockets or binding sites, in other words, can be detected. So let's simplify this surface as a 2D picture. So let's say that so we have this shaped, protein surface. Then as I mentioned, so if some ligand bind to some, convex part, if some ligand, let's say that if ligands comes to here, then they may not have, many interactions. But if some ligand comes into here, then it can have, many interactions with surrounding atoms. Also, so if a ligand comes into this, concave site, then it can have, many interactions. And so here, so that this concave site also can be a, possible protein ligand binding site. So the ligand binding site. So computationally, using computers, people find, possible binding sites by calculating these properties. So that if a probe, so the probe means that, so here the black dots, a probe is inside a concave pocket, then they may have more interactions with neighboring atoms, right? But if a probe is at convex site, they have less interactions with, protein atoms, right? So that is the also another assumption of protein ligand binding site, and we use this property to find or to predict protein ligand binding site of a protein.